I'm Karthik Rajsekharan. I'm the Associate Medical Director at Jazz Pharmaceuticals. And today my talk is going to be slightly different. I'm not a geneticist. I'm not a clinician. You've heard some wonderful talks here, and I've enjoyed it too today, and I've learned a lot. But I can tell you what we are trying to do, and it's a work in process that I'll share, but I'm also trying to focus a little bit more for the caregivers who are here. I think it's wonderful to see your participation. And I think the LGS community is a great example of how you collaborate with researchers and clinicians to advance the science around Lennox-Gastaut syndrome. What's Jazz's interest in studying Lennox-Gastaut syndrome? You all know that there is a product that we are commercializing called cannabidiol. And so we have a lot of participation from the LGS community in the clinical trials. So we have data, and we tried to see what more we can do, and I'll talk a little bit about it. But you're going to see basically I have 10 slides here, and probably I can finish with just 2 slides because you've seen the rest of the slides from other speakers here. Let's look at that. Oh, I was not expecting this. All right, that's okay. But this is also a very beautiful picture. Okay, so this is okay, let's continue, and I'll probably go with this and see where it goes. Maybe there's a slide version here. That's okay. But it is not a single disease. Lennox-Gastaut syndrome is a clinical syndrome which has multiple components and some core features. And a lot of time, what we talk about, and this again is from your website, the LGS Foundation's website, where we do talk about the known causes of LGS, the unknown causes of LGS, 30% of them presumed genetic. But again, even within the known causes, there are these other conditions, like Dr. Grinsman spoke about the infantile spasms. Why do some patients with infantile spasm develop LGS? Why don't everybody develop LGS? Is there potential genetic basis that underlies that? Maybe is there a double hit-like phenomenon where there might be some kind of genetic risk that is underlying that gets unmasked to result in the progression of the disease into a Lennox-Gastaut phenotype. And as an example, I have put this particular slide here, this study, and this study comes from Argentina earlier in the year, where they basically look at children who had infantile spasms, and then they wanted to see what is the genetic etiology. 14 of the 24 children evolved to LGS, and out of which 50% of them had a genetic etiology. So why exactly didn't all of those children progress? Why did some of those children progress, and why do they have a genetic etiology but others do not have it? We don't have answers right now, but it is very much possible that genes probably underlie some of the reasons why this happens. And so there are a constellation of symptoms that probably contribute to the formation of the spectrum, and genetics probably underlies that. So we do have different disease presentations, and probably it underlies treatment response. So this is an example of a study. This is from Canada, where they were looking at the association of trisomy 21 and Lennox-Gastaut syndrome. Apparently, it's infrequent, so you don't see a lot of people with trisomy 21 or Down syndrome having Lennox-Gastaut. But in this particular study, what they found was there was about 10% of the patients who did have Lennox-Gastaut syndrome. But what was also interesting about it is that in those patients, the predominant seizure phenotype was that of myoclonic seizures. So that is what was there for most of those patients. 5 out of the 6 patients had myoclonic seizures, the predominant seizure symptom. And this was very unique in that sense that the authors concluded that, myoclonic seizures are not typically the primary symptom in Lennox-Gastaut. It's more like an associated symptom, but in this instance, it turns out to be the predominant symptom. The other thing that they also spoke about from a treatment response perspective is that they wondered why in their cohort they had a much higher representation of patients evolving into Lennox-Gastaut syndrome. And the rationale that came with it was based on some other studies, and what was also done in Colorado, is that these patients in trisomy 21 infantile spasms are pretty common. And these patients were treated, the majority of them, with vigabatrin. And the idea is that they may have probably responded better to hormonal therapy, ACTH potentially, rather than vigabatrin. And it is quite possible that treatment with vigabatrin could have been the reason why they evolved into Lennox-Gastaut, whereas if they were treated with ACTH, it may not have been the case, right? So again, point being, a genetic underpinning to a disease such as Lennox-Gastaut syndrome, or a syndrome which Lennox-Gastaut is, can also tell us about what drugs to avoid and what drugs may work. So treatment response is critical. And so genetics could help understand a lot of these things. All right, so I had this slide. This is a different version, so I'm kind of trying to figure out in my head, how did I send the wrong one over? But of course, genes are instructions, so I think about a cookbook, then it's a recipe. DNA is the cookbook, genes are the recipe. When I want to cook properly, then I'll take a printout, that's the mRNA, a copy, and then the final product is what comes out of it. But then again, the protein sometimes can be prevented from being produced properly, or the amount of protein that's produced can differ, and how they work can also be affected, their functions. And those are the variants that may underlie, that may contribute to second hits. All right, I'm going to skip past this, and I can tell you what we did in the cannabidiol studies. So this is an ongoing effort. So we had a number of participants in our clinical trial, right? So we did 2 studies, and so we have a total of 396 participants in our RCT. In addition to that, prior to the RCT, we had a compassionate access program, or the expanded access program. And within that cohort, there were about 232 patients who had a diagnosis of LGS. And so we tried to go back to the records and look at the genetic underpinnings that may have been there in these participants. What we found was that the number of participants with a documented variant was 91 in the RCTs. In other words, 1 in every 4. And whereas for EAP, there were only 32 of them. So there was significant diversity in the clinical trial population. Many of them were distinct syndromes, and the word cloud tells you what were all the different gene variants that were identified in the patients who participated in the participants of the studies. And what is also important is many of them did not have a variant noted. Is it because it was not documented in the CRFs? Is it because they did not test themselves for it? And maybe there was a testing, but then, maybe there were genes that were not discovered at that point in time. So there are probably many other reasons, but one of the other ones could also be that the underutilization of genetic testing in adults could have also contributed to it. 1/3 of our population in the RCT were adults. So we do not know the why, but again, one of the things that we can show is that the many different syndromes that were seen within this population. So, what is the biological mechanisms? Some are chromosomal mutations that affect a lot of genes. But then you also have channelopathy, so ion channels not functioning properly, deficiencies in how a synapse is being formed. There could be epigenetic contributions, but some of these genes can underlie structure. So if you have the neurons resulting, going to the regular spaces in the brain, the way they're supposed to, and you have those malformation of cortical development, then you can see problems with the structure. There are also genes that regulate metabolic pathways, respiratory chain. In the mitochondria can be impacted by some of these things. Anyway, too many different phases of LGS. They're all very diverse origins, but then again, the challenges are shared. And what I have put there in the circle are a list of conditions, a list of gene variants, each of which, so many of them actually have their own foundations. So there are many DEEs that basically kind of come inside the umbrella of LGS. And again, like how Dr. Dulugos just mentioned, the EEG may be slightly different, but again, the constellation of symptoms that a lot of these people have are oftentimes similar, and there's a significant overlap between the different conditions, so we can learn from them, and we can also understand more about LGS by learning, also, sharing our information with them. So I think there's an opportunity there. Last but not the least, there are benefits of screening for adults. I'm going to skip because the timer is all the way down. But the benefit of adults here in this example talks about how you have patients, once the testing is done and there is a variant that is discovered, that you might be able to probably make a difference in the treatment that is being provided or the treatment strategy that is being adopted to take care of them. There is a parent perspective, and having a diagnosis, a genetic diagnosis provides relief. It provides an opportunity for the family to really understand what underlies the condition. At the same time, I also think with peer support, like how you have in LGS, it empowers you to become champions for your loved ones. So the key messages here are the causes of LGS are diverse. Different variants contribute to the evolution of the disease. There are many different phases of LGS, but then each phase is a distinct biology. Understanding the genetic underpinnings of the disorder can improve treatment and management. And last but not the least, adult testing is also important. And I think that gaps and opportunities, when I think about it, for future clinical trials, it would be nice if there is documentation of genetic information, either it's there or maybe we can offer genetic diagnosis as a part of the recruitment process, so that if we are able to have that, then at the end of the study, what we can do is we can then probably look at these different subcategories by etiology within this huge bucket of LGS patients, trying to say which patients may have responded better to a treatment versus what was the magnitude of response to a treatment. So all of those things can be done. With that, thanks again. And it's slightly different, but I think I managed it. Thank you.